Okay, I think we're going to go ahead and get started. So first of all, I just really like to welcome you all to the first of our St. Mary's Watershed Initiative webinars that we're completing over the next several months. We get the distinct honor and privilege of going first, and so we're going to give you a little background and some things to think about today. But before we do, we have some important kind of introductions and announcements. So first off, my name is Sarah McMillan. I am a professor in agricultural and biological engineering at Purdue. I am the leader of the water quality team. And together with Mandy Lemiak, who I will let introduce herself. Go ahead, Mandy.
Hi, I'm Mandy Lemiak, and I am the research scientist who's been in charge of doing all the field and lab work for the water quality team.
So we're really excited to be here today to share a little bit about our work. And Ruben Goforth is also presenting today. Go ahead, Ruben.
Hi, I'm Ruben Goforth. I'm an associate professor of aquatic ecology in the Department of Forestry and Natural Resources. The face that you've probably seen out in the field has been Miss Megan Gunn, who has moved on to a different position. So I'm kind of stepping in to do what she's been doing now, but certainly the bulk of the work has been done by Megan, and she's done a great job.
Indeed. We also have several of our broader team here today. So I will just quickly introduce those folks. So Dr. Linda Prokopi is here, who is the lead on the social science component of the project. Linda, are you just waving? Okay. And her research scientist is Emily Usher, who has also been here doing a lot of the coordinating with surveys and interviews. And Sarah Peel, who many of you know well, coordinating education outreach efforts in the watersheds. Dr. Ron Turco having some technical difficulties and will be joining us as soon as he gets those worked out. So without further ado, I'd like to introduce our host for today. So Lindsay, sorry, I muted you on accident. I was trying to spotlight you. There we go. Lindsay Bloom is our host for today. It's a little different when you're hosting a virtual meeting. We're just lamenting that we can't have coffee and donuts for you all, but Lindsay is the resource specialist with the Indiana State Department of Agriculture in Adams and Allen counties, and is kind of hosting this meeting for us today. So thank you so much for participating and helping us kind of coordinate all these things, and really appreciate you being here. Yeah, great. So, and then also we have Ben Wicker, who I will let him introduce himself. Go ahead, Ben.
Hi, everyone. I'm Ben Wicker. I'm the executive director of the Indiana Agriculture Nutrient Alliance, and look forward to the discussion today. Thanks.
Okay. And with that, I think we have all of the introductions completed. I would like to just do a couple super fast housekeeping rules. All of y'all are already muted, which is great. I think most of us have become quite Zoom savvy over the past several months. But please, if you have questions, this is a little difficult or different way of interacting than an in-person meeting, but we do want to try to create some interaction opportunities. So if you have questions that pop up as we go along, please type them in the chat. There's a little button that has a chat with a little kind of cartoon bubble on the top of it. Type your questions in there. You can type them to either myself or Mandy privately, or you can just type them to everyone to generate discussion. And we'll be keeping an eye on that throughout the webinar. If you like to keep your video on, that's great. As a teacher who teaches in this mode, sometimes it's really nice to see people's faces and your reactions, and it feels like you're a little more in the room with us. But also we recognize that bandwidth is a potential problem for any of you, and myself included. So hopefully, if you feel like that's happening, feel free to turn your video off and on whenever you feel like it warrants it. And then last but not least, kind of how we're going to roll this thing today is that we're going to play a series of short videos and then give you some opportunity for interaction and questions in between each of them. So there will be, kind of give you the table of contents, so to speak. We have 3 short videos that we are going to show. Each are about like around 10 minutes long or so, and then we'll have a brief break in between each of the videos to generate some discussion, ask you some questions, hopefully get some questions from y'all. And I see that Dr. Turco just jumped back in. So hopefully his internet is now up and running. Ron is the project lead of the overall projects in the St. Marys and is also the soil health lead as well. And he will be coordinating the upcoming webinars. Ron, anything to add?
Hello, everybody. I'm back.
Great. All right. Without further ado, I will turn the mic over to Mandy, who will get us started.
I think we're planning on starting with a poll. We just want to get to know a little bit more about you guys and what brought you here this morning. So I'm going to throw that up and have everybody just give us an answer. It's totally anonymous, so don't worry about that. And we'll kind of see what we come up with.
Okay, I'm going to end the poll and hopefully this will show you all kind of who's in the room. And so while many of us are working directly with farmers or working with cities, that seems to be the biggest proportion of the group today, there's others who are just curious about what we're doing and passionate about environmental issues and are excited to learn more. So we're excited to have you all here. We recognize that many farmers would love to join us today, but it is very busy harvest time, trying to wrap up everything with a beautiful day. It's important to make sure that all of the work gets done. So we recognize me. It's okay. Technical challenges always around. So thanks so much for participating. That's our first little interaction point. And we'll go ahead and play the next video. Thanks. Oh wait, is it not sharing results? Sorry. There it is now. Can you all see it? Okay, great.
My name is Laura Johnson. I'm a research scientist with the National Center for Water Quality Research at Heidelberg University. My research focuses on nutrient pollution and its impacts on Lake Erie. For 3 days in August 2014, hundreds of thousands of Toledo residents couldn't drink their tap water due to a dangerous outbreak of cyanobacteria, commonly called a harmful algal bloom, in western Lake Erie. We know that harmful algal blooms in Lake Erie are caused by excess dissolved phosphorus in the water. This phosphorus feeds the algae and bacteria so they grow explosively and cause problems for drinking water, fisheries, and other uses of the lake. How do we know that dissolved phosphorus from non-point sources and heavily agricultural watersheds is causing harmful algal blooms? While phosphorus pollution can come from many sources, in this case, the dissolved phosphorus is largely coming from agricultural lands. The Maumee River is mostly agricultural land, which means it's full of non-point sources of phosphorus. A non-point source is often land runoff. Here's why this matters. Pollution that comes from a discrete source like a pipe, point source pollution, gets diluted when it rains. The phosphorus coming out of the pipe mixes with the higher flow in the river, so you've got less phosphorus per gallon of water. But with non-point sources like the water that runs off a farmer's field or a roadway, rain can pick up extra phosphorus from these surfaces and carry it directly into streams and rivers. That means more rain and more water runoff, measured as discharge in blue on this chart, leads to higher concentrations of phosphorus, the orange points, exact opposite of what happens with point source pollution. How do we know which source of phosphorus is contributing the most to Lake Erie's harmful algal blooms? Here's our first piece of evidence. Since the 1970s, pollution from non-point sources has gone up and down consistently. But pollution from point sources has declined sharply and has been fairly steady or even slightly declining since the 1980s. Yet, over the past few years, harmful algal blooms have become a bigger problem than they had been in decades. In 2011, Lake Erie had its biggest on-record outbreak of toxic algae. This photo, from September 3, 2011, shows a massive algal bloom covering the entire western basin of Lake Erie. Yet, on almost the same date the following year, in 2012, there was hardly any toxic algae visible. What made the difference? It's all about the rainfall and the subsequent amount of water runoff or discharge as we measure it. In 2011, we had strong spring rains that caused a massive spike in phosphorus loads from agricultural lands in the Maumee River watershed. In 2012, by contrast, we didn't get the heavy rains, and we didn't get those big phosphorus loads in the Maumee, and Lake Erie was spared a big algal bloom. In 2013 and 14, discharge and load were up again, and both years had larger algal blooms. Let's look at 2014. You can see a number of storm events in the Maumee River from March through June. For each increase in discharge, concentrations of dissolved phosphorus also increased. So, to sum up, watersheds that are primarily characterized by agricultural land use are showing big spikes in phosphorus from non-point pollution after big rains, as indicated in the top chart for the Maumee, compared to no significant spikes in the urban suburban Cuyahoga. Those big phosphorus loads from agricultural watersheds are seen in every year in which Lake Erie has had a major algal outbreak, as indicated in the lower chart showing bloom severity. Where we've had low or no heavy spring rains in an otherwise normal year, like 2012, we've managed to avoid a big algal outbreak. The good news is this. Our research shows that if we slash phosphorus pollution, we don't need to wait years and years to reap the benefits. Decreases in phosphorus pollution will have an immediate impact in the same year. If we can prevent heavy rainfall from carrying phosphorus into nearby streams, the problem would be dramatically reduced. This is a problem we know how to solve. We've already reduced the amount of phosphorus associated with sediment from leaving agricultural fields. If we reduce the amount of dissolved phosphorus that runs off of agricultural lands, we'll make huge strides towards preventing future emergencies like the one that hit Toledo in 2014.
Hi, I'm Ben Wicker, Executive Director of the Indiana Agriculture Nutrient Alliance, a broad collaboration of agricultural and commodity organizations, conservation agencies, and other NGO conservation partners that are dedicated to keeping Indiana's farmers at the forefront of the nutrient management and soil health practices that will improve their farm's viability while ultimately reducing nutrient loss to water. I'm also a certified crop advisor and farmer here in East Central Indiana. Fertilizers and manures are critically important to our farm's long-term viability and sustainability. But as we've heard, excess nutrients in watersheds can cause water quality challenges, particularly in places like the Western Lake Erie Basin, where excess phosphorus and nitrogen cause harmful algal blooms that impact the lake, users of the lake, and others upstream. Increasingly, the general public, consumers, and policymakers are asking questions about where those sources of those nutrients are and what the users of those nutrients are doing to more efficiently manage those resources. Ag is not the only source of nutrients to the watershed, but we do contribute through nutrients dissolved in the water that leaves our fields, and as well as nutrients tied to sediment through erosion and runoff. Managing ag nutrients can be a real challenge, whether it's the weather, access to equipment, time and labor constraints, but voluntary conservation requires all of us to make decisions that minimize the risk of loss to the environment. The good news is that there's benefit to us available on our farms as well. All nutrients have a cost to them, and managing those nutrients more efficiently can add to a farm's bottom line, while preserving our freedom to operate, as well as keeping those management decisions at the field level. It's important that we consider the 4 R's of nutrient management. Are we using the right source of nutrients, applied at the right time, in the right place, and at the right rate, while improving and protecting our farm soil health by minimizing disturbance, increasing our soil's cover, growing more things more often, and increasing the type and diversity of plants that are grown. We don't have to make this journey alone either. There's assistance available out there through our agronomist and our other ag retail. We can look for cost share and technical assistance through our conservation agencies. And there is increasingly all the time amounts of technology and research that point to ways that we can improve the use and the efficiency of nutrients on our farms. By continually improving and evolving the practices that we use on our farm to improve our soil and plant nutrient uptake while minimizing our risk of loss to the environment is what will lead to our mission of healthy soil, clean water, and viable farms. Thanks, and enjoy the rest of this series.
Most of the ground I farm does head to Lake Erie. Actually, all of it does, so I'm in that basin. And after being exposed to the algae blooms that are just over here in Grand Lake St. Marys, or even up in Lake Erie, it got me concerned about what we were doing with phosphorus applications. And being no-till, I thought that was great, that was the right way to do things, but then we find out that surface applications of phosphorus aren't the right way to do it. So that now has morphed into ideas of injecting phosphorus rather than surface applying in the no-till situation. Through history, that was the standard accepted practice. You applied your fertilizer after harvest in the fall. If we were doing tillage, then you worked it in. I went down the no-till path for a lot of reasons, but it saves soil erosion was my first consideration, not knowing anything about fertilizer running off. We just applied it in the fall and had that job done so we could concentrate on planting in the spring.
All right, so here we're at our first little break. I don't see anything popping up in the chat, which is totally fine. But I did want to just get a quick, I'm going to do one poll without the polling feature. And if you have questions, put them in the chat or concerns. I'm curious how everybody's connections are working and if the videos are going okay for you. And if you're having any trouble with seeing them, you can send either me or Mandy or everyone for that matter a chat and let us know. That would be helpful so we can help kind of make sure that things go well today. Okay, I'm going to just do a quick poll based on the things you heard. And this is just to kind of get a little bit of kind of how much of the information that you were presented relating nutrients and agriculture and algal blooms, how do you feel about your understanding? Because it helps us kind of guide the things that we discuss moving forward. And I'm going to give it just a couple more minutes as folks kind of put those answers in. Okay. And I'm going to share these results, and I think it's pretty striking. I'm not sure how much of your the higher understanding came from the things you just saw. It's not our intention really to kind of measure pre-post here, but we just really want to kind of get a sense of your understanding and your kind of familiarity with these issues. And it looks like in this room, we have quite a few folks who work directly with farmers and cities as part of their jobs. And so not surprisingly, the understanding of how nutrients, water quality, agriculture, all these things interact to affect water quality is pretty high. So really appreciate that perspective from everyone. Another question that I was curious about, since we don't have anything specific in the chat, is the conversation and the discussion that both Dr. Johnson, as well as Jean Whitty and Ben, for that matter, discussed about reducing phosphorus and often kind of things related to nitrogen, because actually as part of this project, you'll see data from both. And one of the things that is often a question we get is why phosphorus? Why is it so important compared to nitrogen? Because depending on which basin you're in, those nutrients are differentially targeted for reductions. And oftentimes the targeting practices can be similar, but sometimes they're a little different. So Jean Whitty mentioned about phosphorus applications and how that can be managed through tillage and how that changes the approach. So I just wanted to kind of underscore what Dr. Johnson mentioned about how phosphorus is driving water quality in Lake Erie. This is a little complex biology and ecology, but essentially in freshwater systems, lakes, rivers, kind of inland water bodies, phosphorus is almost, very rarely is phosphorus not the pollutant or an excess nutrient concern. It is the one that is limiting algal growth. So it's the one that if we add more, we get increases in algal production. Conversely, if we go to places like the Gulf of Mexico or Chesapeake Bay, nitrogen is the primary limiting nutrient there. So the more nitrogen that is sent out of the Mississippi River Basin, for example, is what's driving nutrient excess and algal production and eutrophication in the Gulf of Mexico. So those big scale things are different, but in local systems, sediment, E. coli, as well as other pesticides and other kinds of runoff are all kind of important to our local rivers and streams. So it's really important for us to look at both. And so what you'll see in the next series of slides and discussion is this kind of holistic look at water quality as we zoom in to the paired watersheds that we're focusing on in the St. Marys River Basin. And I'm going to open it up to see if there's any questions. And if not, I will go ahead and let Mandy start our next video.
This project is looking to characterize how farm management practices affect ecosystems, soil, and water quality by utilizing education and outreach in the area. For water quality, we're focusing on a few major factors: nitrogen, phosphorus, suspended sediment, and E. coli. These are important water quality factors as they directly affect local stream health and habitat, but also contribute downstream where Lake Erie faces issues of eutrophication and harmful algal blooms. We sample from 12 stream segments and 12 tile outlets in the area on the map, 6 each from Nicholson Creek and the Weber-Gerke Ditch subwatersheds. We sample monthly from April to October at a minimum, usually extending this all year round when winter permits. The USGS installed gauges at 2 of our sites, 1 on each stream, so we now have an extensive timeline of stream discharge. And in conjunction with that, we have installed 2 autosamplers that take daily samples. This allows us to more closely examine storm patterns.
When looking at nutrient concentrations in tiles and streams using our monthly sampling visits, we notice similarities between Nicholson and Weber and Gerke. One key factor is the difference between tiles and streams. Tiles generally have a higher nitrate concentration since they're directly draining ag fields, which often have fertilizer application. But they have lower phosphorus concentrations. These data were collected during our monthly sampling, which covers a range of levels of flow. We're then using statistical models to help us identify any patterns and controls. So what's the takeaway here? The good news is the majority of our samples are showing concentrations below water quality target values. However, these are really complex systems, and while we're learning a great deal about the variability over the annual cycle, we're excited to connect these data with more detailed monitoring we recently started through our partnership with the USGS. Together, the 2 data sets will give us a much fuller picture of what's really driving water quality in these streams. As I mentioned, the USGS partnered with us on this project to install 2 gauges near the outlets on each of our study watersheds. This has allowed us to have a record of discharge starting in August of 2018 with much greater detail than our monthly sampling trips can provide. And we've installed autosamplers to take water samples daily so that we can capture rapid changes in concentration as a function of that stream discharge.
To everyone on the internet. And all you have to do is go to usgs.gov in order to get to the site. And scroll down, you'll see a water category. If we choose the state of Indiana, it will take us here, and if you click anywhere on the state, it will take us to the Water Watch USGS site. Because we know which stream we're looking for, we're going to type in Nicholson. And it will zoom us right in to the town of Decatur, basically, where we see Nicholson and Gerke Ditch, as well as the nearby St. Marys gauge that the USGS has. And we click on one to see it. We'll click on Nicholson and open up its stream flow data. So we'll take you to a summary page. And we want to choose looking at the time series for daily data. That'll take us to this nice graph showing us the last few months worth of discharge data. If we go back, and you can actually zoom in or out here, it'll show us even more of the local gauges that are around, and we'll choose Gerke Ditch now. And go back to that daily data, and we'll see here the last few months of data, and also you can choose which data you'd like to look at. So we're going to change it and go all the way back to 2018, which is when these gauges were installed. And now we can see that even greater data set available here. Another simple way to get to this is to just type in USGS Nicholson Creek into your search bar, and the top result is usually the correct one. And we're looking now at the last week's worth of stage gauge and discharge. So the stage gauge is the stream depth in feet, and the discharge is the amount of water flowing through in cubic feet per second. And again, we can change the date and look at different time periods here. This data is also downloadable. So we go back to the search bar and type in USGS Gerke Ditch, and that will take us to that same top search result, and we'll be able to see the discharge and stage gauge for the last week for the Gerke Ditch.
We'll look first at the Gerke-Weber Ditch USGS streamflow data, along with our autosampler water quality data. Note, this is a small watershed. This only drains approximately 2.6 square miles to where the gauge is located, which is why we see a quick rise in stream flow when it rains. We also see in the top graph that nitrate is the dominating factor in the nitrogen story. Ammonia concentrations are consistently very low. We also see that total phosphorus shows a bit more variability than soluble reactive phosphorus. So we're now going to zoom in on this yellow area where we see a really wet period during the spring of 2019. This spring had a number of large and medium-sized storms in just over a month's time. So to zoom even further in and look at some nutrient data we collected at the same time, we see that there's a nitrate pulse that's happening at the very beginning of the storm as an initial flush, but then is slowly diluted a bit after the flushing is done. Because the rain is continuing and more water is continuously flowing from the watershed, the concentrations decrease after this initial pool is flushed out. We generally think of this nitrate as being held loosely in the unsaturated soils above the tiles. This could be from fertilizer or manure that's applied, as organic components, such as crop residue, manure, as those decompose, the microbes convert ammonia to nitrate and it becomes less sticky. When it rains, water infiltrates into the ground and sends this pool of easily movable nitrate to the stream. Phosphorus, however, behaves a bit differently. We first see a dilution of phosphorus with the initial amount of water, and then it begins to increase and continues to increase with each compounding storm. This pattern suggests that P is more tightly held in soil than nitrate and becomes more of a slow leak rather than a quick flush. Now, switching to look at Nicholson Creek, which has a much larger drainage area of about 13 square miles, we again see the nitrogen portion dominated by nitrate, with spikes in concentration during storm events. Because of the way that nitrate builds up in the soil from the actions of soil microbes, the long dry periods between storms can result in even larger pools in the soil. So we're going to focus on this highlighted time period with some smaller storms following a dry period to see how this plays out in our study watershed. We're going to look more closely at these first two storms that hit following 3 months of a really dry period and very low discharge. So nitrate concentrations are indeed higher than we would have expected during this time, during these smaller storms. And this is similar to what other systems also show following such a long dry period. There's a lot of variability in the total phosphorus, but these storms seem to show a similar pattern as the wet period with a slightly delayed peak.
We've combined both watersheds and separated them into low flow conditions and storm flow events. While there's certainly variability, we see higher concentrations during storms consistently over time. We see this for nutrients, but also for sediments and E. coli. And when we combine these higher concentrations with larger stream and tile flows, we quickly recognize the importance of storms as key drivers of nutrient loss. This is similar to what we see in other agricultural areas throughout the Midwest. To summarize and put this in a slightly different perspective, we'd like to show you a little cartoon of a forest and agricultural landscape. When it rains in a forest, we see a stream flow response, but because trees and shrubs soak up the rain and it infiltrates into the ground, the peak is smooth and low. Nutrient inputs are low as well, and they're rapidly recycled by plants and microbes. In agricultural fields, tillage and tile drainage practices have modified the hydrology, leading to less water storage and quick flow of water and runoff through the pipes into the stream. Nutrient inputs are much greater also as fertilizers and manure are spread on fields for crop growth. Conservation practices have a great potential to reverse these effects. Practices such as cover crops, riparian buffers, and two-stage ditches increase infiltration rates in the field soils and slow the runoff. Nutrient management and modification of phosphorus applications such as knifing into the soil allow more nutrients to be retained in the field for the crop, and that lowers export to the downstream water bodies. Cover crops and no-till practices increase soil organic matter, but they also decrease erosion. Together, these reduce the impacts to our downstream water quality and improve our water quality not only in Lake Erie, but in our local rivers and streams as well. Our work as part of the St. Marys River Watershed Initiative has allowed us to look more closely at the water quality in the Nicholson and Weber-Gerke subwatersheds. We see that during storm events, we have higher concentration of nutrients. And when combined with high flows during these times, it suggests that the majority of our nutrient load is exported during storms. We also measured higher concentrations, particularly of nitrate, during storms that happen after a long dry period. Nitrogen exists primarily as nitrate in these systems, while phosphorus exists both as dissolved reactive phosphorus and a significant component as particulate phosphorus, which is attached to the sediments. We will continue to monitor in-stream concentrations over the coming year, particularly in the spring when we get the majority of our rain and can better understand these storm dynamics. We are also combining this data-driven analysis with a watershed model that will allow us to more closely understand how implementation of conservation practices can be optimized to reduce downstream losses. Together, this integrated look at multiple constituents, nutrients, sediment, and E. coli, provides greater insight into both downstream exports, but also the effects on local environmental conditions. This is particularly important as we seek to improve water quality, habitat, and biodiversity in our local rivers and streams. Okay. That was a little more data intensive. Hope you were able to kind of stay awake through all of that. I have another poll, but before I do so, if there's any questions that you guys have for us, this is a bit of a synthesis of the work that we've been doing for the past couple years. And it's easy to kind of try to pull it together in a way that we think helps kind of tell the story, but we also want to make sure that we aren't leaving any unanswered questions on the table. So I've gotten one question, but I would like to see if there's any others before I answer that. And I will also throw up another poll for everyone to kind of consider. Any questions? If you do have a question, please feel free to just unmute yourself and go for it. Okay, well, hearing none, I'm going to go ahead and ask and hopefully answer the question that was sent in. It says, why is there a difference in between flow and chemistry in the watersheds we see slightly different compared to what was shown by Laura Johnson in the Maumee River. So what Laura showed was this really big rise in phosphorus concentrations with these big storm pulses. What we see in these smaller systems is a much more, I would say, messy situation in some ways. It's a lot more variable. And what happens in these larger systems is there is an aggregation of that variability, or basically things are kind of coming together and you see a concentration and a little bit cleaner look at water quality impacts at a larger scale. When you have these smaller systems, you can imagine that it rains in different kinds of ways in different parts of the field, and flow paths kind of come into the ditches and streams slightly differently and potentially faster in some places and slower in other places. It also affects kind of just the variability by which one farmer is maybe planting corn, another one planting soybeans, another having more conservation practices, and that high variability is kind of magnified when you're looking at a really small scale because there's just a lot more patchiness. So while we're not seeing technically different results, we are seeing considerable export higher during storms than during base flow. And the interesting thing about that is that not only are concentrations higher, we're also getting more water. So even if the concentrations weren't that much higher, we would be getting a considerable amount greater mass or load out because we are taking that concentration and just applying it to a larger volume of water. So the total amount of nutrients being sent during storms is really increased. And like Laura said in her video, we can have direct impact on that year to year, because if we can kind of minimize how much of that pulse of water moves through there very quickly, we can slow that pulse. And what we're seeing is that some of these smaller systems, it leaks out more towards the end of the storm, which is actually good because that's when we have less water moving through. So that is a little bit more of a complicated answer. I hope that wasn't too wordy, but the short story is that we do see similar patterns of storms being really important. And when we zoom in at the local scale, we can see that there's some interesting dynamics about actual timing and processes that are happening that allow us to really better target and understand which practices make sense at the local scale. Okay, and speaking of practices, I'd like to gauge a little bit, we talked a little bit at the end there about practices that do different things and affect water quality and water quantity. I'm curious about your knowledge of these different practices that are part of the IANA strategies and other conservation strategies throughout that you're probably very comfortable and heard about a lot. You may select more than one. It should allow you to do all of them if you're really comfortable doing so. So please go ahead and answer your question, and we'll give a minute to kind of recap after. Sorry, I was muted. Try again. I'll give you just another 30 seconds and then we'll close the poll. Oh, it looks like it only allowed folks to select one option. I thought I had fixed that. I'm so sorry. But I think we'll still get an interesting look at what's happening here. All right, I'm going to end the poll and share the results and recognize that this may be skewed because people selected one option and then tried to do more and probably couldn't. So my guess is that many of you are familiar with many of these, but cover crops was listed first, so it probably got the majority of the votes. As in the chat, or just open up your self to chat and unmute yourself. Are any of these particularly new to you or ones that you have questions about? Because this is really helpful as we think about planning for the future. I see a few folks who are actually the video and shaking their heads. They've got all of these. They're really good. Or you can use, there's a cool little reactions button at the bottom. It has a little smiley face. You can click on that and give me a thumbs up or a heart or, I don't know, a little like crazy celebration icon if you want to. Put a little icon to tell us how comfortable you are with all these things. Looks like most everyone is pretty knowledgeable in this room. Much of the participants are folks who do this as their job every day. So many of these things are probably not new to you, which is not surprising. Okay, with that, I am going to turn it over to Mandy, who will play our third and final video.
My name is Ruben Goforth, and I am an aquatic ecologist and associate professor in the Department of Forestry and Natural Resources at Purdue University. I'm also one of the co-investigators for the St Marys River Watershed Project. My particular interest in this project is focused on the biodiversity of agricultural ditches and how these engineered ecosystems are evaluated for their biological integrity. While they are often viewed as having poor biological integrity, I believe we should think of them differently based on their origin. They are engineered ecosystems with artificial habitats within a landscape context. They therefore do not have the characteristics generally thought of for natural streams. We have to consider that even though they are engineered ecosystems, ditches comprise the representative surface waters of many agricultural landscapes. There are also ponds, lakes, remnant wetlands, and even large rivers present in some of these landscapes as well. But the ag ditches are often the dominant features of freshwaters in agricultural landscapes. It's common for agricultural ditches in the Midwest to score very poorly in terms of their biological integrity. This is because they are generally evaluated based on how they compare biologically and ecologically to what are considered benchmarks for high stream quality. This includes characteristics like fast-running, shallow, clear water that's flowing over rocky stream beds with a substantial leaf fall contribution in autumn. There's no question that these types of characteristics are common and descriptive of many natural streams. But that's not the kind of characteristic that we see associated with these ditches. And in fact, we wouldn't expect to see that at all, because many of these areas were wetlands in pre-European settlement history, and the ditches are not installed to approximate natural streams. They're constructed to remove water from the landscape to allow for agricultural production. Regardless, we evaluate their quality assuming they should perform as natural streams in landscapes where streams were not necessarily present historically. In these European studies, what they've done is they've actually thought about ditches in the context of how they compare to other water bodies that are similar to this ditch that I'm standing in here now. So for example, in this case, there's a lot of vegetation that's overhanging. You can see the grass is down into the water, and that's going to be a lot more similar to, say, a lake or to a wetland. If we start thinking about these as ecosystems with habitats similar to other slow-moving or largely static water bodies in the landscape, as well as the biota that are associated with shallow areas of these water bodies, we can better appreciate the potential positive contributions of agricultural ditches to regional biodiversity conservation, instead of lamenting their poor biological integrity compared to streams that are really not appropriate benchmarks. This will allow us to evaluate them for the biodiversity they are appropriately capable of supporting. So a good bit of what I've been talking about already has alluded to the fact that we don't really think about agricultural ditches as being important for biodiversity in the U.S. And I think that is something we got to work on. In Europe, the work that has been done has been focused on the aquatic insects that live in these kinds of habitats. When I say aquatic insects, I'm referring to both aquatic larval stages of insects that eventually emerge as terrestrial adults, as well as insects that are wholly aquatic throughout their lives. Aquatic insects play multiple roles in agricultural ditch ecosystems. For example, they contribute greatly to processing organic materials in the ditches. This includes leaves and other debris that drops in or blows into the water from outside. They break down these materials for food, and the smaller organic materials that result are then processed by other invertebrates. This promotes the movement and processing of organic material before it is exported downstream. There are also burrowing invertebrates that promote bioturbation and bioirrigation that introduce oxygenated water to buried organic materials so that they can be processed. Finally, they are important in food chains as both predators and prey. This includes serving as important food sources for fishes during aquatic life stages, as well as for terrestrial animals when they emerge as adults. The terrestrial adults serve as food for birds, amphibians, and reptiles, both close to the ditch and in other parts of the landscape. Of course, ditches are also important for a variety of fish species as well, although the full extent of ditch use by fishes is not completely understood. Again, the emphasis has so far been on how fish communities of agricultural ditches compare to benchmark streams, and this type of evaluation consistently scores ditch fish communities as poor. And yet, they still have fish in them. They still have fish that move in and out of these systems. And so what can happen is that these serve as refuges, especially during dry spells when fish congregate in isolated pools until higher flows return. Fish also move around a lot, so fishes in ditches can take advantage of resources such as invertebrate prey when they are abundant, and then move to downstream locations and serve as prey for larger fishes or become recruited to regional populations of those species. One of the habitat types within the ditch that is particularly important is deeper water habitat. So those areas that are more pond-like and have a greater volume of water present tend to have a greater number of aquatic insect taxa and fish as well. And so periodic maintenance to keep the ditches in a form that promotes the biodiversity is going to be important, not only from that biodiversity conservation context, but also because it helps to still promote the rapid transfer of water off the landscape. So I think there are many opportunities to look at agricultural ditches from a biodiversity rich standpoint as opposed to a biodiversity poor potential that I would argue is currently the common approach in the Midwest. I think we have real opportunities for innovation in thinking about how agricultural ditches are evaluated for biodiversity and manage to promote sustainability of both the appropriate biodiversity and the agricultural production needed to support a large and growing world human population.
Hello, I'm Ron Turco. I'm the department head of agronomy at Purdue University and a professor who studies soils and soil biology. I am also very fortunate to be part of the project leadership for the St. Marys effort. The St. Marys effort developed to allow us a better understanding of the interaction between agricultural management and water quality in this critical Northeast region of Indiana. The project is supported via a 319 grant from the Indiana Department of Environmental Management, but we also have excellent support from many in the ag community, including the Indiana Corn Marketing Council, Indiana Soybean Alliance, Indiana Farm Bureau, the Nature Conservancy, the Indiana Ag Nutrient Alliance, and the Indiana Pork, Dairy, and Poultry Associations. We'd also really like to thank all the farm operations in the area who participated in the project. The coronavirus has changed how we do many things, including the delivery of information. The St. Marys project was going on before the epidemic started and will finish before the epidemic is truly over. However, to ensure we convey what we have learned from this project, we have developed a set of online meetings as a way of presenting the information to the St. Marys community. You have just participated in the first of these online efforts on water sciences. In December, we're looking at soil fertility, followed in January by a presentation on soil health, and then cover crops in February, dollars and cents in March, and to close out the series in April, we'll be looking at the 4R program. While we'd hope to present all this information in person, this set of virtual presentations is our best alternative. We look forward to presenting information on our efforts and thank you for your support and participation.
All right, as we wrap things up, I'm gonna send a poll out to you guys to kind of gauge a little bit of interest, but I also would love to hear which topics that you had maybe wanted to hear about or maybe are excited to hear about. You can type anything that you don't kind of feel is poll captured on the poll in the chat as well. And as I launch that poll and you guys have an opportunity to participate, Lindsay would like to share a little bit from the local perspective. And I think she's just going to share her screen. So go ahead, Lindsay, the floor is yours.
All right, everybody. I asked the Soil and Water Conservation District and NRCS if they had anything they wanted to add. NRCS is having their Environmental Quality Incentive Program, EQIP. Their application deadline is for Friday, December 18th. The Conservation Stewardship Program, CSP, their application deadline is Friday, January 8th. And then on your screen, you guys are able to see that the Soil and Water Conservation District is actually hosting a winter conservation meeting December 8th from 10 to noon, which will be held virtually. Jim Foreman is coming, and they'll be talking about profitability of cover crops and making no-till corn to succeed. They have requested for credits. Any questions, you guys can go ahead and call the conservation office at 260-724-4124 extension 3 to reach NRCS or SWCD. So, Sarah, I'll pass it back over to you to finish up.
All right. Thank you so much. I'm just going to leave this screen up in case folks want to grab those numbers or dates or anything, if that's okay.
Perfectly fine.
All right. All right. Looks like everybody has participated in our polls, and I'm going to share results with everyone right now. And it looks like everyone's super jazzed about our upcoming webinar series. Yay. That's exciting. Like Ron said, this is the best of not the best alternatives, right? Like we all wish we could be in person. We all wish that things were a little bit different. We could have a little more engagement with each other. Our approach to this project is always to learn first from your concerns and then help our understanding of our science be better, do better in terms of our data collection, do better in terms of our solution development, that kind of thing. This is a little one way, which is not ideal, but at least we're sharing a little bit of the things we've learned. And with that, I don't think I have any more questions, but I would love for one last poll. We have just a couple minutes before we're at the 10 o'clock hour. This one isn't on the polling, but I would love to hear if you have anything you would like to share in the chat. What is one takeaway, one thing that you learned that you might be able to go back and share either with family and friends who farm in the watershed, family and friends or colleagues or folks you know that don't, maybe people who live in a rural residential home but don't farm? What are some things that you've learned today that you might be able to share with those around you that could make a positive impact on both local water quality as well as water quality downstream? And I just want to say, as you guys are thinking about the answer to that, thank you so much for your participation today. We recognize that your time is super valuable, and especially at this time when there's a lot of demands on your time for both your own jobs, but your family kind of managing the world that we live in. Just really appreciate your participation today. And so with that, I'm going to go ahead and ask, Lindsay, can you stop sharing? Do you mind? I'm going to stop sharing. Bring us all back. Ask everyone to unmute if they would like to, turn on their video so they can see their family, their collaborators in this webinar day. If you're up for that, if you are in your pajamas, feel free to stay muted. I love the commercials that are happening these days of folks dressed up in their, nice top and then sweatpants. It's a great way to have at-home meetings. But it looks like several of you are actually in your offices, which is great. Thank you so much for all of your help. And again, like Ron said, we really appreciate the folks who are our local kind of, folks working with all of these things on the ground. Lindsay, thank you so much for hosting and sharing your information, and everyone who contributed to the videos today. With that, I will capture the chat, so I'm going to leave it open. I don't have to go anywhere just in the next couple minutes, so happy to stay on if folks want to have a little more informal discussion. And yeah, thanks, and have a really, really great rest of your day.
Great job, Sarah, Lindsay, Mandy, Ben, Ruben, everyone. Excellent.
Yeah, it was a lot of fun. We got to do some pretty cool video work, which is a lot more work than you realize sometimes.
A lot more work than you realize.
I hope that everybody's videos turned out okay. If they didn't, we're going to be posting and sharing these on our website, so you can go back and view the webinar. It's in its entirety, but also the little mini clips of the different videos, so they're a little more digestible and easier to share. So we'll go ahead and have those up by the end of the week, probably.
Nice brown work, Ruben.
I promise that mine is actually much smoother and not as pixelated when it's not streaming over this kind of I guess, venue. So if you want to see it in a little bit more sort of real time where everything's matched up, maybe we can see it separate, like Sarah was just saying. But yeah, it was actually a lot of fun.
The video works great on a local drive and even off of YouTube. It's just the multiple things are challenging. You got a lot, there's some just beautiful pictures and a lot going on in them, and it's hard to like render all of that awesomeness in the over the Zoom thing, right? All right, thank you so much, everybody. I have a few things we'll capture in the chat.